Alright, so welcome to everyone that is watching this webinar at the moment. My name is Bram Spruijtenburg. I'm a PhD candidate from the Radboudumc and CWZ Center of Expertise for Mycology in the Netherlands. And I would really like to thank the ECMM and especially Dr. Oliver Cornely for the invitation to give this webinar here today. It's truly an honor to do so. And the topic of today is advances in genotyping techniques. I won't go into too many technical details and I would like to focus instead more on the clinical utility, what can it mean for patient outcomes, and also how can it aid the infection prevention and control measures in your hospital. However, if you do have questions regarding the technical details or any other questions that might arise after listening to this talk, please reach out to me via email. I'm more than happy to answer your questions. And with that being said, I would like to start with the definition of genotyping. What does it actually mean within the field of medical mycology? Well, it basically means to determine the genetic relatedness between isolates, either of the same or different species. And usually when we have genotyping results, we would like to visualize our results. And often we do that in the two ways that you can see on the slides right now. And I will briefly explain them in order to understand the following slides better. So first of all, here we can see a minimum spanning tree. What is important to know is that each dot is a genotype and each part of the dot is an isolate. So for example, in the upper left corner of this figure, you can see a dot that is divided into 4 parts. That means there are 4 isolates with an identical genotype and that are therefore closely related to each other. If you look a little bit more to the right, you can see a lot of small dots that are not divided into parts. So that means that they are isolates with a unique genotype and also not closely related to other isolates from this species. And what is also important to know is that the longer the branch, the more genetic distance isolates have. So for example, up here we again see a lot of long branches. Those isolates are genetically not closely related at all to each other. But if we look more down at the figure, we see all very short branches. And that basically means we have a certain genetic clade that is all closely related to each other. Now, another way to visualize our results is the phylogenetic tree that you can see over here. What is the most important is the horizontal line because that indicates genetic distance. The longer the line is, the less related isolates are. And the vertical line is basically irrelevant. It's only for visualization purposes. So now that we know this, why do we want to do genotyping in the first place? Well, a very important application is to know the population structure of a species, either in your region, in your country, maybe even on a species level. And then we basically are determining the genetic diversity. Do we see that isolates are closely related to each other? Do we see a high genetic diversity? Do they group into clades? And then, when we know this, we can correlate this with, for example, antifungal resistance, a certain geographic distribution, or maybe a certain virulence profile. And well, exactly that is what the authors did in this study over here. This is on Candida tropicalis from a global collection of isolates. And as you can see, overall you see a lot of long branches. That means that genetic diversity is high, but nonetheless there are a few clades of related isolates. And most interestingly is clade 4, because clade 4, as you can see, basically is always resistant to fluconazole. And what they also found is that this clade is highly prevalent in China and also found in India. So, by knowing this, you might change your patient treatment, especially when echinocanins are not available. Fluconazole might not be a good option when you know this clade is prevalent. But of course, the most classic example when we are talking about population structure is Candida auris. With genotyping, we now know that there are 6 genetically distinct clades. And each of those clades, they behave quite differently actually. And here you can see, for example, the prevalence of clade 1 and 4. Clade 1 is highly prevalent in South Asia, while clade 4 is highly prevalent in the Americas. The fluconazole resistance rates differ a lot. Clade 4 is basically always resistant, while in clade 1 is basically always resistant, while clade 4 only half of them. And what we also know is that these two clades, together with clade 3, are associated with outbreaks, while clade 2, 5, and 6 not yet. So if you see in your practice, for example, a clade 2 isolate, the chances for an outbreak are rather low. But if you apply genotyping and you see it's clade 1, you might already take some preventive measures. And then we come to the next topic, and this is basically the most important application of genotyping when we are suspecting a potential outbreak. And I will illustrate that with an example from the Netherlands. What we can see in this bar graph over here are the numbers, is the number of Candida auris cases over the years. It was rather stable, but in the last few years we are seeing an increase of cases, unfortunately. And sometimes we even see that the same hospital has multiple cases close after each other. The question that we have, of course, is, are isolates related to each other? And if so, where is the source? Is the source in the environment or within the hospital, and therefore nosocomial? Because if we do have nosocomial transmission, we need to take infection prevention and control measures to stop the transmission, prevent future patients from getting infected, and also to reduce the burden on the healthcare personnel. And apart from all this, actually controlling an ongoing outbreak is very expensive. It can be a significant financial burden on hospitals as well. So better prevent them and have good surveillance going on. So that's of course what we did in the Netherlands. We applied whole genome sequencing to all of these Candida auris cases, and we can see the results on the right side. Isolates were either allocated to clade 1 or clade 2, the clades that are highly capable of causing outbreaks. But luckily for us, the branches between the isolates were rather long, and those SNPs could not have been accumulated in a very short time. Therefore, we could exclude nosocomial transmission, and we did not need to take any measures to stop an outbreak, for now at least. But unfortunately, outbreaks, especially fungal outbreaks, as you know, is a big problem. We have Candida auris, we have Candida parapsilosis, also common yeast like even the tropicalis is capable of causing an outbreaks. But also even more rare yeast species are causing outbreaks. This is from a recent review that we wrote. And what we see is that outbreaks are occurring all across the globe. They are increasingly reported and very often no source is found. So when you do see multiple cases by rare yeast species, but also by common yeast, it's very important to apply genotyping. And nowadays, when we are discussing genotyping, there are a lot of methods that have been developed. Some of them are still in use, but when discussing these, it's very important to take a few aspects into account. First of all, the resolution. How good is your method to differentiate isolates from each other? Can you actually use it in an outbreak setting, or is the resolution rather low and you are not able to say, are isolates related, yes or no? Of course, the cost is another important aspect, because ideally we would like a low cost, and we can also apply this more often. The turnaround time is another very important factor, because when you are suspecting a potential outbreak, you do not want to wait 2 months to get the results back. No, you want the results ideally in the same day, because then you know if you need to take measures to stop the outbreak, yes or no. And of course, what is important is also the reproducibility. Especially because when you are doing surveillance for a longer time, it's very beneficial that you can compare your current results with results from the past, or maybe even compare your results with other labs. So these are all aspects to take into account when you want to apply genotyping. And now I will discuss a few methods, the most important ones, and that are still in use actually. The first of all is multi-locus sequence typing, abbreviated as MLST. What we do with this method is that that multiple genes are amplified with PCR, and next with Sanger sequencing you get the nucleotide sequences, and those are compared between each other. This method has been used a lot for very good reasons, of course, because it's highly reproducible. You can investigate isolates after a year or even in a different lab, and you will get exactly the same results, so that's excellent. However, for an outbreak setting, this method is not that ideal because it has a very low resolution. So very often you cannot say if isolates are related, and you need something else actually. But for population structure investigations, what I showed earlier, this is a very, very nice method. Another method is amplified fragment length polymorphism, abbreviated as AFLP. A big advantage of this method is that it can be developed in a very short time, especially if you have a new species that you did not encounter yet. You can still apply this. Unfortunately, the reproducibility is rather low, especially between labs. You cannot compare results, and the resolution is variable. In this figure, what we can see over here is that the resolution was actually quite good. That species of Trichophyton in this case were clearly differentiated from each other. However, if you're not that experienced with this technique, the resolution can be quite poor, so that is something to take into account. Another method is the Fourier transform infrared. This is quite a new method actually, and in contrast to the other methods that I discuss and also will discuss, this is a method based on proteins. All the others are molecular methods. What is the big advantage of this method actually is that it has a very short turnaround time and cheap. Two things you really would like in an outbreak setting, but at the moment the reproducibility is rather low actually. Results here, what we see are on Candida auris, and there we actually show that you can differentiate the Candida auris clades quite well, but within clades you cannot really tell if isolates are part of a clonal outbreak. Nonetheless, this is a very new method, so a lot of progress can be made, and maybe in the future this will be one of the methods of choice. So let's see how that works out. Another method that I personally use a lot in my lab is short tandem repeat genotyping. What you need to know is that several of these STR markers are amplified with a PCR, and then capillary gel electrophoresis is performed. This is a highly reproducible method, and usually you can compare results between labs accurately actually. Another big advantage, it has a short turnaround time. If I start my experiment in the morning, in the afternoon, I will already have the results. So that's excellent when you are dealing with an outbreak. But of course, every method also has some downsides, and we also need to be fair on this one. It does require some machinery like a capillary gel electrophoresis, which is especially in low to middle income countries not always available. And you do need some time to develop this method. When you are dealing with a new species, you need to develop the primers, you need to optimize them. So that's a downside, but once it's running, it's very suitable or applicable to outbreak settings. But of course, the golden standard needs to be discussed as well, and that is whole genome sequencing. Why is this the golden standard? Well, by far it has the highest resolution. Even if two isolates differ in a single position, you can differentiate them if you have a good pipeline or good analysis ongoing. So that's excellent actually. However, this method has a long turnover time, especially when you outsource it and when you send your DNA to a company. It might take several weeks before you get the results back, and then you also need to do the analysis yourself. So it's not very great for an outbreak setting. It's also associated with quite high costs actually, but fortunately the turnaround time but also the cost are decreasing every single year, and it's also getting more accessible luckily. And interestingly, you can not only see if isolates are related, but at the same time, you can also have a look and predict antifungal resistance, or take a look at certain virulence markers, what the authors of this study did over here for Candida auris, for example. So WGS, the golden standard, it gives us the most information, but there are some challenges that are luckily getting solved at the moment. So now let's go into a few examples of these outbreak situations or genotyping methods. And of course, the classic example is once again a Candida auris outbreak. This study was conducted between our lab and a single Greek hospital. And what you can see in the lower left corner, actually in red, the red lines, they indicate the colonization. And what we can see is that sometimes the colonization rate was as high as 30 or even 40% and was basically always above 10%. So a lot of these patients, specifically in the ICU, were colonized. And in blue, you can see the number of actual invasive cases. Initially, it were only a few, but over the time, we saw basically always infections, and this is actually still ongoing to date. So the question is, of course, are patients infected in the hospital, or are they already colonized in the community and then they are admitted? So that's why we applied, in this case, STR genotyping. The results can be seen on the right. And what you can see in green, that are the Greek isolates, they all have an identical genotype, except for a single one, but that's from a different hospital. So what we know is that all of these patients are not colonized before. No, in the hospital, they get colonized, and then later on they develop an invasive infection. So there's a lot of work to be done for the infection prevention and control unit to prevent or halt this outbreak. And another application of genotyping, well, not only is about the outbreaks, it's also about the resistance. So an example over here is what we saw with several patients from Kuwait, actually, is that when they were admitted, they had an echinocandin susceptible isolate. Later, they were treated with echinocandins, and very shortly afterwards, the clinicians and the technicians, they isolated an echinocandin-resistant isolate. The question is, what we had, are these patients developing the resistance during the treatment, or were they maybe reinfected or recolonized with an echinocandin-resistant strain? Well, and then we applied whole genome sequencing on these isolates, and the results you can see at the right side. And what you can see is that isolates from the same patient all grouped together, regardless of the echinocandin MICs. So that basically means that all of these patients developed the resistance during the treatment. So this also makes us understand better how a species can adapt even in a very short time. And might also rethink how long should we treat our patients, actually. Another example of genotyping and resistance is this time on Candida tropicalis, because what we saw in the Netherlands was a bimodal MIC distribution for flu cytosine that you can see in the lower left corner, actually. Isolates either had a very low MIC or a very high MIC, and that is rather unexpected because flu cytosine is barely used in clinics and not even in the agriculture in the Netherlands. So that's why we were wondering, are these resistant isolates related to each other, or do they form numerous genotypes? So that's why we applied again this STI genotype. And what we actually saw was that the non-wild type isolates are related to each other, what you can see here in the blue in this phylogenetic tree. They were not clonal, they did not have all the same genotype, so we did not have a single source in the environment or within the hospital. But it does show us that there is a sort of Dutch clade, which is also found in other European countries actually. It is circulating, must be somewhere in the environment, but at the moment we don't know where. But at least this makes us better understanding that this clade is actually present there. So to summarize, I hope that by now you know with those examples that genotyping can give you a lot of information regarding the resistance, and also to know if we have an outbreak, yes or no. Nonetheless, there's a long way to go, because recently we conducted a survey on Candida auris, especially the diagnostics and the screening practices, and we also asked about genotyping. And well, to give you a brief sneak peek into the results, in the left you can see the availability of certain diagnostic techniques. Maltitol was overall quite well represented, although it differed a lot between countries. But when we asked about genotyping, only very few centers could do actually genotyping on Candida auris. Some of them outsourced it, and most of them they didn't. So a lot of effort or a lot of room for improvement to actually um, increase our infection prevention and control. I hope that by now you know that genotyping is essential to confirm or exclude nosocomial transmission. If you have questions, once again, please reach out via email. I'm happy to answer them. And with that, I would like to thank you all for your attention, and also to all of our collaborators who make these genotyping studies possible. Many thanks, and I wish you a nice day. Bye.